Okay, in this segment, I want to talk about other ways to establish pharmacogenomic associations. That is to say, associations between genetic variation and drug response. And the first one, we've already discussed genome-wide association, commonly used, some problems with power, some problems with common variants that are just markers and not the causal variants. Another method I want to talk about is candidate gene approaches. These are making a comeback because of our ability to do cheap and deep DNA sequencing. Cheap and deep DNA sequencing. So in candidate gene approaches, you actually do a little bit of thinking. Remember for GWAS, we didn't have to think about the genes because we were going to test all of them. Candidate gene is, as it implies, you sit and you think about, for example, what you know about the pharmacodynamics of the drug, or what you know about the pharmacokinetics of the drug. And that gives you a set of genes that you think are highly likely to influence the drug response. So this gives you a set of genes. This gives you a different set of genes. And these are not 25,000. These are tens, maybe hundreds of genes, and therefore a much smaller selection of genes to look at. You still need patients with variable response to drugs. But now what we're going to do, for example, is sequence all of these genes. So we take these genes. We have gene 1, gene 2. Let's just say it's 3 genes on this list. Usually it would be more than that, 10, 50. We have maybe 10 to 100 patients with the variable response. We can also get patients without the variable response. So these are the controls. So these are cases at the top. These are controls. And we can sequence the genes. Sequence, 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 sequence for all the cases. Sequence, 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 sequence for the controls. Note, just want to highlight a few things. We are not genotyping here. So we're not just looking at common variation. We're doing a full sequence of the genes. And then we're going to get, as I've talked about in other segments, we're going to get some common variants. I'm going to show the variants. Let's use a different color. Let's use pink. Common variants, rare variants, let's just look at gene 1. I've drawn these in a very particular way. I've drawn these to show that gene 1 has a lot more rare variants in it than gene 1 in the cases versus the controls. So if you really wanted to do this technically, you would come up with a statistic, some kind of measure, to capture the fact that the rare variation in gene 1 is much greater than the rare variation as shown by these little pink tick marks in gene 2. Now, I also want to point out that I have a common variant here and here, and they don't look that different in the common variant section of the gene. There's 3 out of 6 here, 4 out of 6 there, not very different. But in the rest of the gene, and with rare variants that would not typically be on a GWAS tip, you're seeing a clear difference in the mutational spectrum for gene 1 and gene 2, gene 1 in cases and gene 1 in controls. That would give you an indication that, variation in gene 1 seems to be statistically significantly impacting the clinical response that I'm interested in. You look at gene 2, maybe it's a negative. So you see some variation in gene 2, you've seen some variation in gene 1. They don't look that different. Well, gene 2 has variation, but that variation doesn't seem to be important for this drug response. And so on for the rest of the genes. So what's the key idea? We came up with candidates so that we didn't have to sequence the whole genome and didn't have to interpret the whole genome. We picked our genes so that they were highly likely to be involved. We then used full sequencing to look at both common and rare variants. In stuff that I didn't discuss, we came up with statistical tests of the difference between the cases and the controls. There's many, many ways to do this. The most common way is what we call a burden test. In a burden test, you are literally counting rare variants, that is to say minor alleles, in the cases versus the controls. And you're looking at the burden of mutation in cases versus controls. You use the control as the expected, and if the case diverges significantly from the expected, that's evidence that the burden of mutation in this gene is associated with the response. Burden is the simplest. People write tens and hundreds of papers on different tests to pick up these subtle differences. But the idea is the same. Candidate genes, full sequencing, identify genes that seem important, and summarize how you could use the information in a full sequence to perhaps predict the response of interest. That's a candidate gene approach. Second approach that I want to talk about in this segment, that was approach 1, is the general class of approaches that use expression data. Expression data approaches. So I will remind you that in the last 20 years, there's been a revolution in our ability to measure the gene expression levels in cells. So we can now take a population of cells and we can measure the expression level of every gene in the genome. And what we find is that for a particular cell type, liver, kidney, brain, of the 25,000 genes in the genome, a typical cell type has about 5,000 actively expressed genes. There's a big plus minus there. But it's not all 25,000. There are 25,000 genes, but you need some of them to be a liver cell. You need different ones to be a brain cell or a kidney cell. And we can measure their expression levels. So that allows us to do a couple of things. One thing, I'll call it 2A, is a very exciting set of data that were generated in a project called the Connectivity Map Project, or CMAP. And you can PubMed search for these papers. But what they did is for hundreds of drugs, they exposed cell lines to the drugs at different doses. So it's hundreds of drugs at different doses. They exposed them to cell lines, and then they measured expression. And of course, they also measured cell lines without any drugs as the controls. This is a pretty exciting data set if you think about it. Because what it tells you, so now you have gene X. Let's just go through it. You have a certain gene X. You expose it to drug Y at a certain concentration, 10 micromolar. And you see that its expression goes up threefold increase in expression compared to the control. That means that this drug at this concentration is doing something to the cell that the cell perceives and decides to upregulate gene X. For many genes, you give drug Y and nothing happens. They would have no expression change. So the list of genes that get changed, so it might be gene X, Y, and Z, which all have this change. These are the genes that are sensitive to the presence of drug Y. Now, Is this the mechanism of action of Y? Probably not. Y gets into the cell, it binds its target, but upon binding that target, it's affecting that target and the pathway. The pathway probably gets probably goes down in activity. The cell perceives that the activity is down, and it says, I'm going to upregulate some genes in response to this perturbation. So this is like a second-order effect of the drug, but it's incredibly useful for understanding how the drug changes the state of the cell. Because there's the direct effect of the drug on its target, but then there's the secondary effects on the overall gene expression. So how can we use this? One way people use this is in an incredibly simplistic but fascinating, is they say, well, look, I have a disease that has gene A, B, and C down, and X, Y, and Z up. If you give me a drug, so that's the disease. It's a very simplified representation of the disease. It's like, it's a cancer cell, for example, and A, B, and C are down, X, Y, Z are up. From the CMAP data set, I have a drug that just happens to make A, B, and C go up, and X, Y, and Z go down. So in an amazing bit of simplistic thinking, we say, hey, maybe this drug will treat this disease because it will tend to normalize the expression level of all these genes. And people have tried that, and people have had success with that. So now we've associated the drug to the entire disease through the expression changes. It's a very different way of thinking about drugs because we're not really thinking about the target itself. We're thinking about the systemic effects on the cell of giving this drug. But we can use this to describe the drug response as expression changes and match up drug and disease responses to try to have them corrected by the drug. So that's one application of connectivity map. And I should say connectivity map was sufficiently successful that there's a follow-on project called LINKS that is measuring even more drugs on even more cell lines at different doses. So it's just a scaled up version of the connectivity map. There's one other concept in expression that I want to discuss. This would be 2B. And this is the idea of an EQTL, which I want to define for you. When we look at SNPs, we find out that there are some SNPs. So here's a SNP that's a GT SNP. And we look at many, many individuals in their genomes, and we find this SNP. And we also do expression of their cells, say expression of their lymphocytes. So we have SNPs. We have 1 million SNPs, actually. 1 million SNPs. This is one example. And we have 25,000 gene expression measurements. And we have this for hundreds to thousands of people. I don't want to confuse it. Simple setup. We have patients for whom we measure their SNP chip to get their genotypes, and we measure the gene expression, say, in their white blood cells. I choose white blood cells because you can get them from blood. You don't have to grab tissue that the patient might otherwise be attached to. White blood cells are replenishable. So white blood cells. When you get this data, you can find something called expression EQTLs, which are SNPs where the allele of the SNP, so remember the SNP alleles are going to be GG, GT, and TT, where these are predictive of the gene expression level. So let's say GG predicts high expression of gene A. GT is normal expression, we'll put normal in quotes, of gene A, and TT is low expression of gene A. There's a bunch of statistical things you have to do, but you can do this. If you find such a SNP, then that SNP is an expression, quantitative trait loci. So let me decode this for you because it's a little confusing. Loci, we know what a loci is. It's the SNP. The SNP is predicting a quantitative trait. That is to say, a trait that's not 0 or 1, but takes on a real valued number. And that real valued number is the expression level of the gene, because that can go from low to high. So an EQTL, or expression quantitative trait loci, is simply a SNP that predicts the expression of a gene. Why is this interesting for pharmacogenomics? Well, sometimes we might have an association between a SNP and a phenotype and a drug phenotype. It might come out of a candidate gene study. It might come out of a GWAS. But when we look at the coding regions around that SNP, we don't see any good genes where we see an amino acid change or an obvious thing that explains if this SNP is causative or simply is in LD with the causative SNP. One mechanism by which a SNP that is otherwise difficult to explain how it could impact drug response, one way it could work is that it changes the expression of a gene that is very important for the response to that drug. So if that SNP, if it's an EQTL for a gene that's in the PK or PD pathway for the drug of interest, then that might give you your causal mechanism. I'll put a question there because it's a maybe. The idea is this SNP, which is sitting not in a gene but out in the middle of the intergenic region, might actually be the causal gene because it is affecting the transcriptional decision about whether to make the gene or not. And therefore, it is causal, but it's not causal through effects on the protein amino acid sequence, but it's causal through its effect on expression of the gene. So just to draw a picture, there's the gene. We usually expect SNPs here in the coding region that perhaps cause a change in amino acid. Usually when we see a SNP out in an intergenic region, we are very suspicious that it's causal. But it is possible that this is in a region, for example, for transcription factor binding, that actually does make it causal because it affects the ability of the cell to transcribe this gene. It still could be an LD with another SNP that's an EQTL, but you can then find all the EQTLs and do an analysis to see which one is the most likely to be causative based on its location in the genome, perhaps its location within a DNA sequence that is the recognition sequence for this transcription factor. So expression data is a powerful and orthogonal source for learning about the genetics of drug response. In the first case, we used it to understand how a drug may alter gene expression. In the second case, we're using expression data to come up with mechanisms whereby non-coding SNPs might affect the expression of a gene in a pharmacokinetic or pharmacodynamic pathway.